Good morning. In this lecture, we will study about antibodies. Antibodies are antigen binding glycoproteins present on B cell membranes and secreted by plasma cells. Those which are bound to B cell membranes gives antigenic specificity to B cells, while those which are secreted by the plasma cells circulate in blood and function as effectors of humoral immunity. Antibodies produced by clones of a single B cell are monoclonal, but antibodies produced in response to a particular antigen are polyclonal because most antigens are complex and contain many different epitopes. The protein part of antibody is made up of 4 polypeptide chains. It contains 2 identical light chains of molecular weight of about 25,000 Dalton and 2 identical heavy chains having molecular weight 50,000 Daltons. Each light chain is bound to a heavy chain by a disulfide bond and non-covalent interactions like salt linkages, hydrogen bonds, hydrophobic bonds to form a heterodimer. Similar linkages link 2 identical heavy and light chain combinations to each other to form a basic 4-chain antibody structure, that is a dimer of dimers. The exact number and precise position of these disulfide bonds differs among antibody classes and subclasses. Further, antibody molecule has 2 regions, variable region and constant region. Variable region, as the name suggests, has amino acid sequence which are highly variable among antibodies of different specificity. Around 110 or so amino acids of amino terminal region of the light chain or heavy chain constitutes variable region. Then there are regions in these variable regions called complementarity determining regions or CDRs, where most of these variations or differences are found. It is these CDRs on both light and heavy chains that constitute the antigen binding site of the antibody molecule. In the rest of the molecule, very less differences are seen in amino acid sequence in a single class of antibody. These regions of relatively constant sequence beyond the variable regions are known as constant regions. Antibodies are glycoproteins. The carbohydrate component of the antibody is bound to the constant region. The role played by glycosylation of antibody is not clearly understood, but it is believed that it probably increases the solubility of the molecules. The knowledge about the basic structure of antibody was derived from a variety of experimental observations. Example: digestion of immunoglobulin G antibody with enzyme pepin produces 3 fragments, 2 identical fragments and a third fragment which is quite different. Identical fragments have antigen binding sites and are known as FAB fragments or fragment antigen binding. The other fragment has no antigen binding site, and since this fragment crystallizes in cold storage, therefore it is called FC fragment, that is fragment crystallizable. Then digestion with pepsin enzyme generates only a single fragment composed of 2 FAB-like fragments. FC is not produced because it is digested into multiple fragments. Now, if antibody is treated with mercaptoethanol, all 4 polypeptide chains are separated because it leaves the disulfide bonds. Now, if we see the fine structure of antibody, we come to know that both heavy and light chains of antibody contains homologous units called domains, which are about 110 amino acids long. Within each domain, and an intra-chain disulfide bond is present, which forms a loop of about 60 amino acids. Light chain contains 1 variable domain and 1 constant domain, and heavy chain contains 1 variable domain and 3 or 4 constant domains depending on antibody class. Then there is a flexible region called hinge region in a heavy chain of antibody, which contains an extended peptide sequence rich in proline between the first and second constant domains of the heavy chain. Due to this hinge region, FAB arms of antibody can assume various angles to each other when antigen is bound. Now let's study about the various classes of antibodies and their biological activities. There are 5 classes of antibodies: IgG, IgM, IgA, IgE and IgD. They differ from each other by unique amino acid sequences in the constant regions of their heavy chains. Immunoglobulin G is the most abundant among all the classes of antibodies. 80% of total serum immunoglobulin is IgG. It has 4 subclasses: IgG1, G2, G3 and G4. These antibodies can cross placenta and provide passive immunity to developing fetus. They are also effective complement activators. Then IgM, immunoglobulin M. 5 to 10% of total serum constitutes immunoglobulin M. It is a membrane-bound antibody on B cells. It is secreted by plasma cells as a pentamer, in which 5 monomer units are held together by disulfide bonds. Each pentamer also contains an additional Fc-linked polypeptide called the J chain, which is linked to the carboxyl terminal of the 2 heavy chains by disulfide bond. It is the first immunoglobulin class produced in primary response to an antigen. It is more efficient than IgG at neutralizing antigen particles and activating complement system because of its pentameric structure. It does not diffuse well due to its large size. Therefore, it is found in very low concentration in intercellular tissue fluid. Then immunoglobulin A. It constitutes 10 to 15% of total immunoglobulin in serum, but it is a predominant antibody class in external secretions such as breast milk, saliva, mucus of digestive tracts. It mainly exists as monomer, but external secretions contains a dimer, a J chain polypeptide and a polypeptide chain called secretory component. Daily production of this antibody class is greater than any other antibody class. IgA secreting plasma cells are concentrated along mucous membrane surfaces because these are the main entry sites for most of the pathogenic organisms. IgE is present in serum in very low concentration because it has very potent biological activity. IgE antibodies are responsible for immediate hypersensitivity reactions in our body like hay fever, asthma, anaphylactic shock. It binds to the Fc receptors on membranes of basophils and tissue mast cells, and on binding to antigen results in degranulation of these cells and thus results in allergy. The last class of immunoglobulin is IgD. It is present in very low concentration in serum. Like immunoglobulin M, it is also a membrane-bound antibody expressed by mature B cells, but its role in physiology of B cells is not known.